Through in silico sequence typing and genome comparison, we were able to compare 5 E. coli genomes as well as a reference genome, MG1655. The first in silico analysis used was multi-locus sequence typing, MLST, which helps track and identify DNA sequence variations and locates unique alleles. Based on the 7-gene analysis, we can see that the sequence types varied for each genome. For the 7 genes, alleles at each locus were assigned according to differences in nucleotide sequences. For example, there were similarities between MG1655 and ESCVA2346, as they both had similar allele assignments at ADK, FUMC, GYRB, ICD, and MDH. Similarly, ESC3A CA3512 and FDAAR-GOS433 had the same allele assignments at ADK and RECA. The close relationship between different genome allele sequences may provide insight into evolutionary history and nucleotide divergence. O and H antigen in silico serotyping is especially important because of the high number of E. coli serotypes that exist. The O and H antigens can be found in nature in many combinations, and this serotype method allows us to target flagellar antigens, which may allow us to detect different E. coli serotypes. Consequently, their results of analysis portray a relationship between U00963 and ESCVA2346. The flagellar sequence located for both were at FLIC H48 with H antigen and O antigen at WZX016 and WZY016 respectively. These O and H antigen types may provide markers into the type of E. coli that exists and variants between different genomes. Analysis of antibiotic-resistant genes and virulence genes from each E. coli strain lead to patterns of differences of strains in terms of resistant gene expression. Many of the strains produce similar gene results, including MDF-A and all. Genomes such as FDAAR-GOS433, ESCVA2346, and ESCCA3512 exhibited high amounts of antibiotic-resistant genes. Virulence genes are important in determining the pathogenicity of an infective agent. The virulence analysis provided similarities with many strains containing GAD. U0096.3 and ESCVA2346 had high similarities for virulence, serotype, and MLST, which may lead us to the conclusion that they're both almost identical and are very relative. The Artemis comparison tool makes it possible to identify regions of similarity, insertions, and rearrangements at any level from the whole genome to base pair difference. Through this analysis, we're able to compare the MG1655 reference genome against the other 5 genes. There seems to be a high relationship between the MG1655 and ESCVA2346 at 99% throughout the entirety of the comparison. Brig analysis gives us a visualization of the relationship between the 5 genes and MG1655. When comparing the reference genome to the other 5, we could see the fewest white gaps would exist in the ESCVA2346, which means they would have the highest sequence matches relatively. Another Brig analysis was done on another strain of E. coli, BA403, and this one had a interesting relationship between ESC CA3512 in that they were almost identical.